So the question we're asking here in this case study is, given all available mutation data, including the latest data we're pulling from TCGA, what are the most frequent oncogenic mutations, i.e. targetable mutations, in a particular cancer type? So I think what we're witnessing is these sequencing studies are identifying a whole number of mutated genes. These mutations could be knockout or tumor suppressor mutations. They could be passenger mutations. But those mutations that we're most interested in are novel oncogenic mutations that are activating a protein and are potentially amenable to therapeutic intervention. So what I'm showing you here, much like what we saw for HER2, I'm showing you the mutation spectrum of two different genes, BRAF in melanoma and a gene called Titin in ovarian cancer. And I would just draw your attention to these very much contrasting mutational spectrum. BRAF, which we of course know is an oncogenic mutation that's amenable to therapeutic intervention, is mutated almost exclusively at one particular amino acid. Other oncogenes like KRAS or NRAS or EGFR also tend to be mutated at hotspots. In some cases, it's a handful of hotspots that are mutated. In contrast, the Titin gene, which happens to be one of the largest genes in the genome and has been shown to be mutated in ovarian cancer, when you take a look at the mutations, what you see is the mutations occur at a whole wide spectrum of the Titin protein. No particular hotspots, and also no particular enrichment for knockout mutations, such as frame shifts or truncating mutations. So it's really, when we're thinking about new therapeutic targets, we're much more interested in hotspot mutations specifically, like BRAF and melanoma. So what I'm going to show you now is the mutation browser power tool and our efforts to characterize oncogenic mutations and tumor suppressor mutations amidst a background of passenger mutations. So what I'm showing you here is a summary of all of the mutation data in the database. So data from COSMIC, from TCGA, etc. Each circle here is a gene that has been reported to be mutated, and we're characterizing a few things about each gene in this plot. So on the x-axis here, we're looking at the percent of mutations in the gene that occurred at a hotspot, or one of the three most common sites of mutation. What we're showing on the y-axis is the percentage of mutations that were clearly deleterious, meaning they resulted in an out-of-frame frame shift mutation or introduced a stop codon, a so-called truncating or nonsense mutation. And what we've found is, when we look at mutations this way, what we can see is that mutations tend either to occur at hotspots or frequently be deleterious. A handful of genes appear to be both, so need to be characterized further. But a whole number of genes fall out of the analysis because they don't show either of these two patterns, hotspots or recurrent deleterious mutations. Now, interestingly, if we look at genes that have frequent hotspot mutations, what we recognize here are a number of well-known genes, but also some new genes that are emerging from sequencing studies. I'll point out that our criteria for showing up in the plot is only that a mutation occurred 3 times at the same amino acid site. If we increase this to 5 times, now we get down to a smaller set of mutations that are always occurring at hotspots in a number of well-known oncogenes, AKT1, HRAS, NRAS, BRAF, KRAS, etc. Even moving down the plot here, what we find are a whole number of both well-known oncogenes and oncogenes that are emerging from sequencing studies. We've now become convinced that this is actually one of the most important axes to analyze and consider mutations on, the frequency of hotspot mutations. I'll point out EZH2. This is a gene that's just showed up recently in this plot. 6 months or a year ago, it wasn't here, and that's because hotspot mutations have now been observed in lymphomas, suggesting EZH2 is an oncogene in lymphomas. Similarly, up here on the deleterious side, what we see are a number of well-known tumor suppressor genes, as well as novel potential tumor suppressor genes that are emerging from next-gen sequencing studies, such as PIK3R1. I want to point out that we can look at oncogene status, so predicted oncogene or tumor suppressor, across all data in the database. We can also look at just particular cancer types. So for example, if I click on colorectal cancer, now we're summarizing only mutation data from colorectal cancers and asking which genes show recurrent hotspot mutations at particular amino acids in colorectal cancer. And because we don't have large collections of colorectal next-gen sequencing studies yet in the database, you can see that it's a relatively small number of predicted oncogenes in colorectal cancer, well-known genes. But we expect that views and analyses like this will become more rich as we bring in more sequencing data. Let's look at brain cancers. So we do have sequencing data from the TCGA on brain cancer, so this plot is informed by that latest data. And if I select the genes here now that show recurrent hotspot mutations, even if rare. So this plot here is summarizing the frequency of hotspot mutations out of all mutations. So even if rare, we believe that these are the predicted oncogenes and potential proteins that could be targeted in brain cancers based on recurrent hotspots. Now in the table below here, what we're also providing are the total number of cases, the number of mutations. So you see for EGFR here, it's less than 10% of all brain cancers have an EGFR mutation. But among these 90 mutations, 48 of these mutations are occurring at one of three of the most common hotspots. Hence the prediction that EGFR is an oncogene in brain cancers. So now what we're going to do is go and look at these oncogenes of interest in glioblastoma specifically to understand their mutational spectrum and their frequency in brain cancers. So now I've just tabbed over in the mutation browser. So we were at the oncogene tumor suppressor tab. I've now preloaded the by genes tab with the genes that we identified as likely oncogenes in brain cancer. Started first by looking at ELK. We see 1,000 cases tested for ELK, 74 mutations. This is out of all brain cancers. But when we drill in on ELK and look at specific brain cancer types, you can see that all of these mutations have been observed in neuroblastoma. No ELK mutations observed in the 91 TCGA glioblastoma cases. Contrast that with a predicted oncogene, IDH1, and I think if you followed the IDH1 story, you'll agree with me that the biology at play here is fascinating, but it's looking like there is an oncogenic aspect to IDH1 mutation. So 34% of all brain cancers, if we look at specific cancer types, yes, recurrent mutations in glioblastoma, so 136 out of 1,400 cases, 9.5%, but much more frequent alterations in oligodendroglioma and astrocytoma. And what we can see is that, again, all of these mutations are hotspot mutations occurring at the 132 amino acid. We could investigate other oncogenes here, for example, KRAS, and ask the question, at what frequency is KRAS mutated in particular cancer types, and do we see hotspot mutations in KRAS?